today, Andy and myself will co-host this seminar, and I will start today's seminar by acknowledging the traditional owners of the land on which Sydney University is located, the Gadigal people of the Eora Nation, and pay respect to the knowledge embedded forever within the Aboriginal custodian of the country. And for those of you joining us online, I would also like to acknowledge the traditional custodian of the various lands on which you join us today, and pay respect to all elders past, present, and emerging. I'll hand it over to Andy for the introduction.
Yeah, thank you, Jean. So it's my great pleasure to introduce Dr. Mingxuan Tsai. So Dr. Tsai is an assistant professor at the Department of Biostatistics at the City University of Hong Kong. He obtained his PhD degree from the Hong Kong University of Science and Technology in 2022, and his broad area of interest lies in statistical machine learning and data science with applications in genetics and genomics data analysis. And today we're very lucky to have him join us to talk to us about leveraging genetic diversity to find map causal variants of complex traits. So I'll hand over to you, Mingxuan.
All right, thank you, Andy, for your introduction, and thanks, Professor Yang, for your invitation. It's my honor to join this Sydney Statistical Mathematics Seminar Series. So today I'm very glad to introduce one of my recent work in by mapping causal variants of complex traits by leveraging genetic diversity. Well, this is a collaborating work with Professor Yang Can from Hong Kong UST and some other lab members from his lab. So I will start with some short introduction of the background. And basically I'll introduce the data structures, the problem we are interested in, and then I will go to the challenges we are facing in this area. And then I will introduce our XMAP model, which leverages cross-population data by leveraging genetic diversity and can help us improve the fine mapping results. Okay, so lastly, I will show you some empirical results, including simulations and real data analysis. And I will also discuss some potential future work, which my group is now exploring. Okay, so first let's look at the data set. Okay, so for GWAS, genome-wide association study, we usually have this kind of data structure. We get some trait vector, which can be either complex phenotype or some expression quantitative trait or proteomic quantitative traits. And at the same time, we get some genotype matrices here. And what we want to do is to find out the relationship between the phenotype and the genotype here. Okay, so the SNP is single nucleotide polymorphism, and Z1 to ZK here is some covariates, which can be potentially some confounding factors that need to be accounted for when we do GWAS. Right. Okay, so typically, traditionally, what GWAS do is very simple. We just marginally regress the phenotype vector to each column of the SNP. We try to find out whether there is a marginal statistical correlation between the SNP and the phenotype. Okay, so if we do this to screen across the genome, what we get is something like this, which is called Manhattan plot. We get x-axis as the genomic position in the genome, and the y-axis is the negative log 10 of the p-values. Okay, so you can see that across the genome, we can actually identify many kind of associations, statistical associations, similarly past some very small p-value threshold, usually 10 to the minus 5 or 8. Okay. So up to the March of this year, actually GWAS has been very successful in the sense of number of associations identified. For example, from the GWAS catalog, we can see that up to this month, we get more than 500,000 top associations, which are very significant. Okay, so this number seems very promising that we identify many genetic signals underlying the human phenotype. But the question is that with so many associations to be identified, it actually makes the downstream biological investigation very complicated. So you get a very very large number of signals across the genome, then for biologists, they will need to follow up for each of the identified signal. That will be very hard and very costly. So, and actually, not all of these statistical associations identified with GWAS are actually causal signals. Now, the first reason is that from this figure, we can see that in the very short local region, like in this green bar, for a very small region, we can have many or multiple SNPs that have statistically significant association with the phenotype. Okay, so if we zoom in, for example, zoom in this area, it will be like this figure. Okay, so this dashed line is a very small p-value threshold. For example, 10 to the power negative 5. But you can see that even in a very small region, we will see that many SNPs, all of them have very small p-values. Then it will be hard to say which SNP is actually responsible for this signal cluster. And this is because in this very single region of the genome, all SNPs can be highly correlated. And this is because in the meiosis of the human, of the cells, the genome is recombined homogeneously in a single population. So if our data is from a homogeneous population, then for single locus, all of the SNPs here can be highly correlated, and this correlation pattern can be quite similar across different individuals. So if we just focus on the single population, the samples from a single population, it can be hard to identify which SNP is actually causal, because the causal SNP can be correlated with non-causal SNPs. Okay, and here this heat map just shows the correlation pattern between among the SNPs in the local region. So all of them can be very high in the short region. So now the key problem is, how can we prioritize the actual causal SNPs from the highly correlated variants in the local region? And this is what biomapping is trying to do. Okay. So as we can see here, we cannot just use the marginal regression to find an association. That does not tell you the causal relationship. So what we need to do is to take this correlation or LD, linkage disequilibrium matrix, into account, so as to more robustly prioritize the causal SNPs in this region. Okay, so if we build that problem into a statistical probabilistic model, it is something like this. We get some phenotypic vector Y, we get the genotype matrix X and its effects B on the phenotype Y. Okay, so X collects all the SNPs in this local region, and B is its effect on the phenotype Y, and we add some random noise here. Now, what we need to do is to identify the non-zero entries in this P vector B. Okay, so in order to do that, we can introduce some probabilistic framework, like we create some random variables called gamma j and collect all gamma j into a vector gamma of P dense. So gamma would be a binary vector with its element being 1, suggesting the SNPs at that position being causal to the phenotype Y, and gamma j equal to 0, suggesting the SNP j has no effect on the phenotype. Okay, now if we can introduce some proper distributional assumptions on gamma and B, then we have a prior distribution P gamma B. Then combine this prior distribution with our probabilistic model here, we can do Bayesian inference. By evaluating the posterior probability of gamma j equal to 1, and which is called the posterior inclusion probability, because it's given the data Y and X and try to evaluate the posterior probability of a SNP j being causal to the phenotype. Now, with this posterior inclusion probability, or PIP for short, we can more robustly rank the causal SNPs or prioritize the causal SNPs by taking the correlations embedded in X into account. Okay. So once we get, now suppose that we have a set of putative causal SNPs. For example, we get PIPs for all the P SNPs, and then we can set up a threshold, for example, 0.8 or 0.9. So that the SNPs with PIP higher than that values, we consider them as putative causal SNPs. With this set of putative causal SNPs, they're actually a purified and denoised set of causal SNPs compared to the GWAS p-values. So with this set of putative causal SNPs, we can do some more reliable downstream analysis. But because this kind of putative causal SNPs will be a much smaller set of SNPs compared to genome-wide significant SNPs, and that carry more biologically meaningful signals. So we can use this purified SNPs to do some downstream analysis. For example, we can combine them with single cell dataset to trying to find out some enrichment in some specific cell types. In such a way, we can try to identify the cellular context of causal variants. For example, we have a set of putative causal SNPs, and we look at its nearby regions and try to identify the genes in its nearby region and look at the gene expression in the single cell RNA-seq data and trying to see whether the set of genes are enriched in specific cell types. Okay, so in such a way, we can identify cellular context of the causal variants. Okay, we can also try to co-localize the SNPs with EQTL data. For example, if we identify causal SNPs of a complex trait, we try to look at EQTL data and do a find map again and see whether two causal signals are co-localized. If it is, then perhaps that SNP is affecting the phenotype by regulating the corresponding gene. In such a way, we can identify the regulatory activity of causal variants. Okay, so this is what we can do with the find mapping output. Okay, so here's a more recent application. Basically, they combine find mapping results with the putative technique. So the first thing is that they try to use the find map, applying it to GWAS data and identify the putative causal SNP set. With this small set of putative causal SNPs, they have just a small number of candidates variants, and then they can do some CRISPR perturbation or do some variant editing techniques and try to see whether this kind of perturbation can affect the downstream gene expression so as to verify the causal relationship. Okay, and they actually found that this kind of find map outcome are indeed influencing the downstream gene expression, and verify the find mapping results is biologically meaningful. Okay, so given the promise of find mapping, we can do many things, but there are still some outstanding challenges that need to be addressed. So the first one is the one, as I mentioned, the linkage disequilibrium among the local SNPs. Okay, so let me use these examples to demonstrate this one. Okay, now this is the data set from UK Biobank, low density lipoprotein, which is correlated with cardiovascular disease. We get the sample size with 300,000. Now, if we look at this local region, we will see that there are a bunch of SNPs. All of them have very small p-values. So blue line is 10 to the power -6. The red line is 5 times 10 to the power -8. Okay, so you can see that all the SNPs here in this region, they have a p-value smaller than 10 to the power -6, which are very small, indicating strong significance. And if you apply SUSY, which is a very widely applied find mapping techniques in GWAS, you will see that this will be the results. Now the y-axis is the PIP obtained from SUSY. Okay, you can see that all of these red SNPs, and include this RS90776, they have just very minor PIPs. The highest one is only about 0.1 something, and almost all of them have PIP smaller than 0.1. So it's very hard to say which variant is actually causal to the phenotype, at least the region. Although we can say that there is at least one causal, because all of the p-values are very small. But because all of these SNPs are highly correlated, simply using this European data set, although we have a very large sample size, we cannot reliably detect or prioritize the causal SNP. Okay, so how can we address this issue? The opportunity comes from the cross-population data. Okay, so this one is the European data I showed you before. In the middle, we get another GWAS data from the African population collected by the Global Lipids Genomic Consortium. You can see a clear difference between these two panels. Okay, so you can see that in Africa, we don't have many SNPs being very significant, but we only get this RS90776 passing the 10 to negative 6 p-value threshold. And that is because in Africa, the correlation pattern among the local SNP is very different from the one from the UK biobank, the European samples. We can figure this out by looking at this third panel. Okay, so the upper triangle shows you the correlation between among the SNPs for European population. So this one, this RS90776, actually is highly correlated with all these red SNPs here. But if you look at the African population, the pattern is very different. You can see that it only has minor correlation with these red SNPs, which are originally highly correlated with it in the European population. Sorry. So this gives us a hint that perhaps we can try to combine the samples from African and European, and then try to conduct joint file mapping and improve the resolution and the power of file mapping. Okay, then how to do that is the one problem that we try to resolve. Okay, now here comes our second challenge, which is the unadjusted confounding effects. So as I have mentioned in the GWAS data, we have phenotype, we have genotype. Sometimes we get covariates to be adjusted. The covariates are usually population stratifications that can confound the GWAS results. For example, the people from the North China are usually higher than the people, systematically higher than the people from the South China. And if we do not account for this population stratification, there may be some inflation in GWAS results. Okay, so here's an example. If we look at, suppose that we are looking at two alleles, allele A and allele B. Okay, now, and we assume that we get two subpopulations, subpopulation 1 and subpopulation 2. Now, if we do not observe the subpopulations indicated here and just look at the overall population, and then we try to figure out the distribution of allele A and allele B in case and controls, and try to compute the odds ratio. That will be 7 multiplied 12 divided by 4 multiplied 9. And this value is 2.3 something, which indicates the major effects of allele A. However, if we just look at the subpopulations, for example, for subpopulation 2, down here, so the dashed line separates the subpopulation 1 and subpopulation 2. If we look at subpopulation 2 only, then we can compute the odds ratio again. Now, here the odds ratio is 1 multiplied 12 divided by 10, which is only 1.2. If we look at subpopulation 2, it's only just 1. Okay, which indicates there's no difference between the distribution of allele A and allele B in case and controls. Okay, now this is just because that if we do not observe the confounding factor here, we have the subpopulation as the confounder. If we do not observe this kind of confounder, just look at the overall, then the results can be distorted. Okay, but sometimes this kind of confounding factors are not observed. So then how to adjust for confounding effects that is not observed and not adjusted in GWAS summary statistics to avoid false positive? That will be a second problem. Okay, so the third one comes from the computational perspective. Okay, so this is a paper from Nature Reviews Genetics. So previously, for traditional fine mapping methods, people usually need to assume there is a specific number of causal signals, causal SNPs in the local region. For example, they need to assume there's 1 or 2 causal SNPs, and then apply the method to identify the SNPs that is most likely to be responsible for this number of causal signals. However, the traditional fine mapping methods are computationally very intensive. They basically need to use exhaustive search to find out which SNPs is responsible for a causal signal. So in that case, the computational cost will be exponentially grow with the number of causal variants you specify. However, this paper just say that usually in the local region, there can be more than 1 or there can be more than 3 or 4 variants in a very short region. So in that case, if you want to identify all of the variants, that will be very difficult because there's a computational bottleneck for traditional fine mapping methods. So how to identify multiple causal signals that coexist in a single locus is a computational challenge. Okay. All right. So let me introduce some related methods. So the first batch are methods that use a single population data. FineMap and DAPG, they use heuristic to improve the computational speed. And SUSY is very widely used recently. It's proposed in 2020 in GISSB and extended to summary statistics in 2022. Okay, and SUSY-INF is published in NG this January. Okay, and the second batch of methods use cross-population data to improve the resolution of fine mapping. So basically, Panther is extended from single population methods, and the cross-population method is published in 2015. However, recent discoveries found that Panther can be inflated because it does not treat the uncertainty in GWAS summary statistics properly. And MS Caviar is proposed in 2021, and it tried to combine different populations, but because it used exhaustive search, so the computation is a problem. So SUSY-X is proposed almost the same time as our method. It's now in bioarchive and is published. Is submitted to bioarchive in 2023. Okay, now let me briefly introduce our model. So our method is published in Nature Communications last October, and it's a collaboration work with Professor Yang Can from Hong Kong UST. Chen Gang is from a Beijing company, and we have other authors: Hu Xianghong, Xiashun Xiao, and Zhiwei Wang from the UST lab. Okay. So this is our model. So basically, our target is to leverage the genetic diversity from different populations. So here we try to include. We use 2 populations to demonstrate. Say we get European as the first model, and another population, for example, African or East Asian, from the second population. So what we do is that we first separate the effects into 2. The first one is a major causal effects, which we use B to represent. And the second one is phi, which represent the polygenic effects. We do this decomposition because we believe that the polygenic effects, which is usually small, may not be that biologically interesting. But the sparse causal effects can have larger effect sizes, and that can be more replicable across data sets and be more biologically meaningful. So our focus is on B part. Okay, now let me first introduce our sparse causal effects. So for population 1, B1, it is a vector of P elements, where similar it is the sum of K causal effects, which is borrowing the idea from Suzy. Okay, so similar to, we have K indicator vector gamma K, each of which is a P vector with values 0 or 1, and each vector contains only one element being 1 and all other values being 0. So the position of element 1 represents the index of causal SNPs of signal K. And then we multiply this vector with beta 1K, which is scalar representing the scale of the effect size. Then we sum across all the K causal signals, we get B1. Okay. Now for B2, we have almost the same structure. We have the same K causal signals and its own effect size beta 2K. But with similar, the causal status gamma K is the same across the two populations, so as to leverage the information from X1 and X2. Okay, in such a way, we can use the difference in LD that is embedded in X1 and X2 to identify the non-zero entries in gamma K. All right, so here are some probabilistic specification for gamma and beta. So basically, we're similar, beta 1K and beta 2K can be different, but their causal status are the same. Right, now for dense part, for polygenic part, we just use the normal distribution to capture that. So here, we get only the individual level data. Now we can extend the model to the summary level data. Basically, for summary level data, we have the marginal effect size, B1 hat, and its associated standard error as 1 hat. These are marginal effect size from marginal regressions, and we also have the effect size and standard error for population 2. And with some algorithmic, we can see that the model can be extended to the following one. So basically what we need is only the marginal effect size B, the standard error S hat. So this S1 hat is a diagonal matrix with its diagonal elements collecting all the standard errors of the P SNPs. And additionally, we need a correlation matrix computed from the genotype X here. Okay, so basically you can see that this summary level model is just the true effect size multiplies the correlation vector, correlation matrix for the mean, and for the variance it's just the marginal standard error reweighted by the correlation matrix. Okay, so what we need is just the marginal effect size, correlation matrix, and the standard errors. We do not need X and Y anymore. Okay, so up to now, we have assumed that the covariates have been well adjusted, but in practice, that may not be the case. So here we consider a way to adjust or correct for the confounding bias in the GWAS summary statistics that is not that will affect our result. Okay, so our idea is based on LD score genetic drift model. You can see that under the genetic drift model, we can extend the summary model further to the right-hand side. The only difference is that for right-hand side, we multiply a constant C in front of the variance term. Okay, so if C is very close to 1, then the two models are very similar, approximately the same. That means there's no confounding effects in the effects in the GWAS summary statistics. However, if C is larger than 1, you can see that the variance will be enlarged in the model of the right-hand side. Okay, that means the original standard error S1 hat here may not be enough to capture the noise in the GWAS effects. Okay, so in that case, we need to zoom in. Well, we need to enlarge this variance term. Okay, so this C in practice can be estimated from data. Usually, when there is no confounding, C will be estimated to be very close to 1. If there is confounding in the GWAS summary statistics, and C will be estimated larger than 1, and plug in this C estimate, it can help us to adjust for the unadjusted confounding effect. Okay, now we put this final model with our probabilistic specification here. Our final goal will be to evaluate the posterior probability of gamma J. Before that, we need to estimate the unknown parameters C1, C2, omega, and sigma K. Okay, so there will be an identifiability problem if we simultaneously estimate all of these, because this sigma K and omega can be identifiable. So what we do is that we first apply LD score regression to estimate omega, C1, and C2 with the genome-wide summary statistic. And then we fix all these 3 parameters in the model and use variational EM algorithm to estimate sigma K. In such a way, we estimate all the model parameters, and then we can finally get the PIP value. Okay, and the reason that we need to use genome-wide summary statistic is that if we just use the local SNP, the estimate of omega, C1, C2 can be unreliable, because we usually have hundreds or just thousands of SNPs, and that value can have high variance. So here's a comparison. The red box is the estimate of omega using SUSY-IN, and which use local SNPs. And our method use the sign box, XMAP. Our method use the genome-wide summary statistics. You can see that our method actually yield much smaller variance for the estimate of omega. Okay, so here's the comparison of computational time. So left figure shows the number of causal variance against the computational time. So red lines are methods. Basically, we are just linear to the number of causal variance thanks to the sum of single effects model specification and variational EM algorithm. Okay, and our model is also, the computational time of our method is also linear to the number of candidate variance, which is the P in the local region. Okay. So here I show you some empirical results from our studies. So for simulation, we consider 500 SNPs in chromosome 22, and the sample size is 20,000 European with 10,000 to 20,000 East Asian. And we set the true number of causal signals to be 3. And we select these 3 signals so that they are less correlated in the East Asian, but highly correlated with non-causal SNPs in the European population, right? Now we first evaluate the calibration. The rightmost figure shows the XMAP's false discovery control. The x-axis is the expected FDR level, and the y-axis is the empirical FDR level. Okay, so we compare across all methods, but the key message here is that XMAP is well calibrated in the case of no confounding bias. And we also achieve the highest statistical power in terms of identifying the causal signals. Okay, now let us consider the confounding effect. Okay, what we do is that we use PC to represent some confounding covariates. Basically, we simulate the phenotype by including PCs when generating data, but when constructing the summary statistics, we do not include PC. In that case, the summary statistics will include some unadjusted confounding bias. And we actually evaluated LD score intercept, which are the C1 and C2 I previously introduced. You can see that as the sample size increase, the LD score intercept increase. That means with the sample size increasing, if we do not adjust for confounding bias, then the inflation will become severe. So here's the FDR control. So the second to the last is the XMAP with the summary statistic that already account for the PC. In that case, XMAP is well calibrated, and this is a ground truth that we compared with. In practice, the confounding covariates may not be observed, so will not be adjusted in the marginal regression. So this one, the last one, shows us the case when the GWAS summary statistics is not adjusted from confounding bias. XMAP has slightly inflation here, but almost very close to the best case, and performs much better than other methods. Okay. So here is a concrete example from simulation. This one is the GWAS from East Asian, GWAS from European. So we can see that for GWAS from European, the red dot represents the true causal. I only use one true causal so as to avoid the so as to demonstration. So we can see that at this corner there are some non-causal SNPs still have very small p-values. But if we just apply by mapping to this data set, you will see that there's an inflation here. These SNPs, they are not causal, but because of the unadjusted confounding bias, they have high PIP, right? And if we combine these two without adjusting for that, for example, in SUSY-X and XMAP, where we enforce the C1 and C2 to be 1, you will see that there's a high PIP here, false positive. But if we just estimate C from the data and reweight the variance term, we'll see that this part is repressed. So the false positive disappear. Okay, so let's go back to our previous example of LDL. This figure shows you the GWAS I showed you before. Okay, this is the GWAS. This will be a result of fine mapping with 300,000 European samples. Now, if we increase the sample size, simply by increasing the sample size, we can manage to improve the PIP of this RS90776 to 0.6. Okay, already very high, but not perfect because it is still hard to say that this SNP is definitely causal because the probability is only 0.6. But if we just use about 100,000 African samples, we can achieve the PIP of 0.9 with single population data. If we use XMAP to combine 300,000 European samples and 100,000 African samples, we can further improve this PIP to 0.99, almost sure that this SNP is causal. Okay. Now, we also conduct some replication study to see whether results of XMAP is reproducible across different data sets. Okay. So what we do is that we use two training data which can potentially be contaminated by some confounding effects with inflation constant 1.6 and 1.12. We try to identify putative causal SNPs and look at their PIPs obtained from another data which has a small inflation constant, which suggests there is absence of confounding. So this third sample is from a siblingship study, because we know that siblingship study is more immune to population stratification. Okay. Now the result is here. We can look at the left panel where we use 0.8 as a threshold to identify the causal signals. So basically, we categorize the SNP into 4 groups. The deep green suggests that those SNPs has a very high, larger than 0.9 PIP in the third cohort. And the light green shows the PIP between 0.5 to 0.9, and deep blue shows you the PIP of 0.1 to 0.5. So we can look at these 3 clusters. If all these 3 clusters takes a large proportion, that means that method can the putative causal SNPs identified by that method is more replicable. Okay, so we can see that the XMAP actually has a larger replication rate here compared to other methods. Okay, and lastly, we try to apply our method to identify the cellular context of the blood trace, because blood trace is there are many single cell data of the blood cells. So it's easier to verify the reliability of fine map results with blood trace. Okay, so we apply XMAP to a bunch of blood trace and try to obtain their corresponding putative causal SNPs and look at the enrichment of that set of putative causal SNPs in different cell types. Okay. And we actually find out the result is quite promising. For example, the red blood cells, for red blood cells, we found that most of the traits are enriched in the erythroid cells. For example, like in this part, all these traits are related to red blood cells, and actually found significant enrichment for erythroid cells. And for white blood traits, we actually found that they are enriched in the lymphoid progenitors. Okay. So this is our method, XMAP. So our group is now actually exploring some further directions in fine mapping. For example, previously we have only looked at the overall level of enrichment for a given trait. But what we do next is to try to find out the enrichment of cellular contacts for individual signal cluster. And what fine mapping can help us to do is more. Like, for example, we can leverage a fine map to help the Mendelian randomization in drug target validation. The drug target validation basically focuses on a single region of the gene expression and try to find out the causal relationship between the gene expression of proteomics quantity and the complex trait. Okay, but the difficulty is that the instrumental variables in this cis Mendelian randomization are usually highly correlated, and we can actually apply fine mapping techniques to help us more robustly identify the valid instrumental variables and improve CSMR. And the last one is to integrate with the perturb-seq data for EQTL fine mapping. So our paper is focusing on complex trait fine mapping, but for EQTL, there's also critical problems, because a recent paper just published last year, they mentioned that basically the EQTL, the lack of localization between the EQTL and the complex traits is because the natural selection. Sometimes our genotypes are just have very rare mutations. In that case, it will be very hard to detect the causal signals. But with the perturb-seq data, which introduce some manually induced perturbations that can help us to improve the power to identify causal signals. Okay, so here are some directions that our group is exploring. And so that is all of my talk. And here is the reference. If you have any questions about either XMAP or the further directions here, you are welcome to discuss with me. Okay, thank you.
Yeah, thank you very much, Dr. Tsai. Do we have any questions for Mingxuan? I can start off. So yeah, I think this is such a cool method. Yeah, thank you so much for the talk. Yeah, I think it's so amazing that like you're taking advantage of genetic diversity, where like most people say it's like a limitation in this sort of field. So my question was, in your method, do you have to manually specify what the two populations are, or because I'm assuming we would have to do it in a manual way? Like, is there a way that you automatically identify like populations to use?
Yeah, that's true. So oops, sorry. Could you see the screen?
Yeah.
Yeah. Right. So basically, what we do here is that we need to know which population, which data set is from which population, because when we deal with summary data, we need to match the correlation matrix with the GWAS summary statistics. And in that case, for example, we know that the GWAS summary statistics like B1 hat and this S1 hat from European, then we need to match the correlation or the LD matrix here, so that this R1 is representing exactly the correlation in this X1. Okay, so when we do modeling, we need to know which data set is from which population, and so that we can do we can pair up the summary statistics and the LD matrix. And this is one. And actually, so when we combine populations from a combined data set from different populations, we can combine more than 2. So here I use 2 populations as an example. But actually, if you have like, for example, sometimes you have samples from many populations, European, African, East Asian, South Asian. Our software is actually adaptable to any number of populations. So as long as you specify correctly the population, you can correctly specify the populations, then our method can leverage any number of data sets from different populations. Yeah, but yes, we need to know we need to know what population it is. Okay, so that's one. Yeah, let's hope this answer your question.
Okay, I think in the interest of time, it's been really a fantastic talk. It's wonderful. We might close the formal part of this session, and then we'll move into a casual conversation. So for those people who have more questions for Mingxuan, please feel free to stay behind and that and then turn your video on.